Hello everyone. Now today we're doing another review on a BGCSE biology paper, and we're doing a paper 3, and this is from the year 2015. Now as usual, please remember, write your school number, your candidate number, your surname, and your initials. Again, read through the instructions carefully and make sure that you follow all the instructions given during the examination. And also remember, do not open a booklet until you're told to do so. Now let's jump into question number 1. And for question number 1, we said the picture shows various sites that can be affected by nearby water supply. And so study the diagram carefully. You will see a construction site, you see a suburban development, you see the city, you see a factory, you see a sewage treatment plant, you see a point source. And the point source is where dumped or substances is being discharged into the water body. For example, the sewage treatment plant, you notice that it's being dumped in the water. And then you have a clear cut, which is the cutting of land. And then you have a farmland on the top right here. All right, so now we observe the diagram and we understand what is going on there. Let's jump into our first question. Now, the first question asks us to define the term pollution. And pollution is a process. So let me write that real quickly. So this is the process by which waste change or disturb the natural balance of the environment. All right, so once the natural balance have been changed or disturbed of the environment, that is actually pollution. All right, and just remember that pollutants are the substances that will cause pollution. And so our next question here is said, name the type of pollution caused by X or in area X. So let's go to the diagram to see what is happening on area X. And area X is the smoke from the factory, which is this point right here. So the smoke from the factory is area X. Okay, so make sure you observe that and see it clearly. All right, let me put an highlight on that for you real quick. So this is the smoke right here coming from the factory. Okay. All right, so let's jump into the answers. And I know you surely know what that is. And that is air pollution. So the type of pollution there is air pollution. Okay. All right, so great. Air pollution. All right, and so the next part of the question state that sulfur dioxide is one pollutant that may be produced in area X. So again, sulfur dioxide may come from the smoke from the factory. It's a give two harmful effects of this pollutant on the environment. And so on the environment, one, it could cause acid rain. So one is acid rain. And you can know the effects of acid rain. So let's put that one real quick. The acid rain. And we know the dangers of acid rain, right? And so the acid rain could damage crops. Let me just do this real fast. So we could have a damage of crops. Okay. We can also have nutrients depletion of soil. Okay. So soil being depleted of nutrients. Okay, so that's that one there from acid rain. And again, acid rain could cause a number of different things. All right, it could cause buildings to be damaged as well, corrosion in certain substances. The next one is you may have respiratory problems or disorder. So respiratory problems or disorder. All right, so there we have, those are the major things that will cause from sulfur dioxide. All right, so let's go to the next part of the question. And the next part of the question state, it reads that name one site shown in the picture that could be a source of pollutants that could cause eutrophication. And remember what eutrophication is. Eutrophication is the excess growth of algae. So let's look at any of these sites that could cause eutrophication. Let's go back to the diagram. And again, what you're looking for is any of the sites that could actually discharge any form of nutrients in the water. So there are two main ones that is seen right here. So we can see here, we have the sewage treatment plant, which is this point right here. And we also have the farm. All right, so the farm, new fertilizers coming from the farm. And even the factory, because it's a point source right here again, because the factories will have, like, for example, bathroom and so on. And so the sewer system could end up depositing waste product within the water body, okay? And so the main two right here, though, is the sewage treatment plant and also the farmland. So let's put that in real quickly. And they asked us to name one, but again, you know, I like to put more than one, so at least you know a lot of knowledge or a good amount of the content, so you can able to answer other questions as well. Okay, so farmland is one of those, and the other will be the sewage treatment plant. Okay, so let's put it on this side. Again, they say one, so you're going to choose one. All right, so this is just for us to practice at this time. So sewage treatment plant. Okay. All right, so those are the two main ones. And again, the factory could also be another one because you have a point source there. All right, so for part two, it's a name the pollutant produced by the site you have named in which could cause eutrophication. And so if you're looking at the farmland will be fertilizer, okay? So if you're looking at the farmland, it'll be fertilizer. And if you're looking at the sewage treatment plant, then that will be the sewage. And of course, the factory will be the same thing, sewage from the factory as well. Okay? All right, so good. And so remember, I'm just doing this like this. So farmland is for fertilizer, sewage treatment plant is sewage is a pollutant. All right? So let's look at now the other question. It said, describe and explain the process of eutrophication. Now remember what eutrophication is. Eutrophication is the excess growth of algae when nutrients are deposited in water bodies. For example, river, lakes, and so on, all right? All right, so the first point I want to mention right here to talk about how it occurs is that nutrients dissolve in water, okay? So nutrients dissolved in the water as a first point. Then the next point you want to look here, and remember this is a 4-point question, so you want to look at the next point right here. Then what will happen here is that the algae will absorb the nutrients. Algae will absorb the nutrients and grow rapidly. Okay, let me kind of go real fast and grow rapidly. All right. And next thing that will happen here after that takes place is that the excess growth of algae, so the next thing I want to talk about here, the excess growth of algae, all right, will cause less light to enter the water or to penetrate the water to, let's put penetrate the water. All right, and as a result of this, then because of the less light, then you have decrease in food sources and so on, right? And so less water will penetrate the water, and the water becomes, and because of this also less oxygen is going in, so what you'll have is the water becomes stagnated. Okay, so next point I'm about here, that the water becomes stagnated, okay, and fill with bacteria, okay? So to fill with bacteria, a lot of bacteria will be in the water at this point, and also this will result in low oxygen, okay? So this will result, resulting in less oxygen. All right, so less oxygen will be in the water. All right, and because of less oxygen, then what will happen here is that you have a lot of organisms will be unable to obtain oxygen for their respiration, so they will lack energy for one, they will have more possible diseases, and also reduction in food sources. All right, so let's jump on to the other part of the question. Well, the next question, because this is finished right here. The next question is that the diagram shows part of a nephron. And remember what nephrons are. Nephrons, they are the filtering units of the kidney, or the functional unit of the kidneys, right? Let's quickly do some labeling. I'm going to label. This is the tubule of the kidney or the nephron, the tubule of the nephron. All right, so in fact, this will be where the Boseman capsule is. So right here in particular will be Boseman capsule. Okay, so let's just put this right here. So this is what they call a glomerulus. So that's the glomerulus. Here we'll have the Boseman capsule. So let's put it right here. All right. E is a little bit more than the Boseman capsule because it's going into the tubule. So I would not label E as the Boseman capsule. Okay. So this is a Boseman capsule. Okay. And then for part C, structure C actually, that is the epithelium or the epithelial cells. Okay, so this is the epithelium. All right, or the epithelial cells. All right, for B, B is going in, so this is coming from the renal artery, so label this as the renal artery. Okay, and A is coming out, so this will be the renal vein. Let's put this as the renal vein. All right, so those are the labelings right here. Let's see what they're asking us to do now. And for part A, it's name the parts labeled B, C, and D. And of course, you have them right here already, so there's no need to go back over that. So we have B, which is the renal artery. C is the epithelium, and D, let's see what D is. D is the, let's go up a little bit, glomerulus, okay? So right, just make a note of that. I will not label them right now just to save some time. Okay, so those are the labelings there. Now for part B, it said explain why the tube labeled B is wider in diameter than the one labeled A. So looking for the wider in diameter. So why B is wider in diameter. Now, I know you'd have wondering what exactly you're talking about, because remember that veins, the lumen, which is inside, is normally wider than the artery. So this question, you have to be careful what they're actually asking you. What they're asking is the entire blood vessel. In other words, why this blood vessel is bigger and thicker, in other words, right? They're talking about the entire diameter from the end to the end, not the internal part, which we call the lumen. So do not be confused with this question at all, okay? Because you would have already known that the lumen of the vein is wider compared to artery. So what they're asking us now is the entire diameter, okay? The overall structure. So for B now, why B is wider, it is simply because of thicker muscle. So one point here, it has a thicker muscle. So let's say this is due to thicker muscular wall. So thicker muscular walls. All right, let's put that there. Due to thicker muscular wall that are adapted to transport blood on the higher pressure, okay, or on the high pressure, to transport blood on the high pressure. All right, great. And so for the 2 marks, you're talking about the thickness of it and the function of it, which is to transport blood under higher pressure. Okay, so the wall is thicker, much more muscle on the walls. All right, so for part C now, it said name the fluid leaving the structure shown at point E. So at point E, which is below the Boseman capsule, which is this point right here, it said name the fluid that is found there. Now remember from the Boseman capsule, a matter of fact, what is coming in the Boseman capsule is the substances that were filtered from the glomerulus. And so this here is called the glomerular filtrate. So the fluid that is found there is called the glomerular filtrate. Okay, so that's the name of the substance, glomerular filtrate. All right, and that's one point there. Let's go to the next part of the question. And this part of the question here is state that glucose and amino acids present in fluid E are reabsorbed back into the blood from the nephron. So it is present in fluid E, which is in the glomerular filtrate, or the filtrate that is coming from the glomerulus. And so glucose is found there, but now it's asking you now how they are reabsorbed back in the blood from the nephron itself. Okay, so let's talk about that. All right, so the first thing you need to put right here is that they diffuse across the walls of the tubules, right? So the process that's taking place is a diffusion process, right? So they diffuse. So how they are reabsorbed, they diffuse or move across. You can say diffuse or move across, whichever one. Okay, diffuse or move across the walls of the tubules. All right, of the tubules, then enter the blood or enter the capillaries and enter the capillaries. And enter the capillaries that surround the nephron. All right, so that's how it actually works. Okay, so again, they will be diffused across the walls of the tubules and enter into the capillaries that surround the nephron. And a point to note is that the nephron will have some convolutions and also loop of Henle in terms of allowing these substances to stay in there longer, and the longer they stay there, the more effective they can be absorbed. All right, so for part 2 right here, it says suggest what will happen to these materials after they are reabsorbed back into the blood. So glucose. When glucose is reabsorbed, and it's a 1 point for each of them. So when glucose is reabsorbed in the blood, one of two things can happen, okay? So one, it can use as energy. It can use as energy by the cell, use as energy, or to produce energy. Let me say use to produce energy, use to produce energy by the cells, okay? All right, so that's one way it can be used, or otherwise, what could happen to it again is that I'm gonna give a next point right here that you can use as well. So if there is an excess, okay, so if in excess, all right, it will be converted to glycogen and store in the liver. All right, so that's another point right there. But again, the primary function here is that when glucose are taken into the blood, they will be sent to the cells. The cells will use them to produce energy by the process of respiration. So they use in the process of respiration. Otherwise, in excess, they'll be converted to glycogen where it is stored in the liver. Now for amino acids, primarily, again, amino acids are used to produce proteins, okay? So here, this is used for protein production, used to produce proteins, okay? It's used to produce proteins. And then let me just put the other concept here. What if amino acid is in excess, okay? So I'm just giving this extra piece here. If in excess, all right, the amino acid will convert to urea by deamination. So deamination will take place and it will convert into urea, and this is done in the liver, okay, in the liver. All right. So deamination can take place and it will convert into urea by the liver or in the liver, okay? All right, so this question ends here. I know this is 10 marks. All right, so let's go on to the next question quickly. All right, so next question now. This one reads that the enzyme catalase found in potato tissue speed up the breakdown of hydrogen peroxide in oxygen and water. Now, the effect of catalase concentration on the rate of oxygen production was investigated by a group of students. Just to stick a pin right here. Catalase is an enzyme that is found in living tissues. So it could be plant or animal. So it could be in liver, okay? It could be in the skin, in the muscles in the skin, and in the body, and so on, right? So again, it is found in living tissues. Now, again, catalase used to break down hydrogen peroxide. All right, so this is a chart right here. I think this is everything. No. The table shows the result. Okay, yeah, this is everything now. So it starts from Concentration being 0, that means no catalase, then there's no oxygen produced, and it's slightly increased, going up by 5, and then the rate of oxygen production increases as well. And the rate is calculated in milliliters per second, okay? All right, so now look at the questions that they asked for this. It said enzymes are described as biological catalysts, okay? And then it said explain the meaning of this term. So what is meant to be biological catalyst? Let me point out something real quick for you, right? To be biological, first and foremost, bio means living, okay? So the word bio comes from living. So biological is referring to living or inside of a living. And catalase is a chemical that used to speed up chemical reaction, right? So you put this all together, we simply mean that enzymes, they are chemicals used to speed up chemical reactions, all right? So enzymes, they are chemicals. And of course, we know what chemicals they are. And in fact, they are proteins, okay? So they are chemical proteins. I could put protein in the bracket right here just to give that extra information here. So enzymes are proteins, are chemicals used to speed up chemical reactions, okay? Will speed up chemical reaction in the body without being used up or changed, without being used up, okay? Without being used up or changed. So the enzyme themselves remain unchanged or being destroyed by the chemical reactions. Say they only use to facilitate the reaction, okay? And again, they work in the body. That is important because it's biological. They work in living organisms. That's where the word biological comes from. Catalyst just simply means to speed up chemical reaction, right? So merging the two, they work in the body to speed up chemical reaction. So simple that, okay? They work in the body or work in living organisms to speed up chemical reaction. All right, for the next part of the question here now, this is a graph here. Let me put a zoom into this. All right, so let me do this real quick. And so I'll do the graph for you, so that saves us some time. Let's go up a little bit more. Okay, so this is the graph. They asked us to draw the graph. Okay, so they asked us to draw the graph. So let's do this real quick. So first and foremost, I said use the data from the table to draw the graph. So already, to save some time, I've done this for you. But let me just point out a few things real quick before I move on, right? Now, when you're doing a graph, there are a number of things you must observe. Please, you must label your axis. So your y-axis, x-axis, please label them. Do not leave them unlabeled. Again, your scaling must also be appropriate in that your graph should not be too small, okay? And you use fine pencil to make your markings. Unfortunately, I can't draw straight with my free hand, so the lines are a little bit raggedy. But I mean, in the exam, you use a ruler and you use your fine point pencil to get these points accurately. All right. Anyway, let's jump on to the question to sort this out real quick. Now, for this part of the question, it said describe the relationship between catalase concentration and the rate of oxygen production as shown on your graph. So if you look at the graph carefully, you notice that as one is increasing, the other one is increasing. The table also shows you that. You do not need a graph to look at to know that relationship, right? Because you notice as the concentration goes up, then the rate of reaction also goes up. All right, so let's write what the relationship is. And so let's put it right here. Let's get back a zoom into this. All right, so here. All right, so quickly, it said here as the concentration. So as the concentration of catalase increase, the rate at which oxygen is being produced also increase. Okay. All right. So that will be our answer there. That's the relationship. So as the concentration of catalase increases, okay, the rate of or the rate at which oxygen is being produced also increase. Okay, so as the rate of catalase increases, then we also have an increase. Catalase increases, the rate of which oxygen is being produced also increase. Okay, all right, so we're good at that part. All right, so let's jump to the next part right here. It said, what concentration of catalase will result in a rate of oxygen production of 2.2 ml? So we want to find out the concentration of catalase when we have 2.2 ml of oxygen. So let's go to the graph and see how we could map that out real quick. All right, so let's follow this now. I'm going to use a bold line really to kind of map it. So 2.2 is right here. Okay, I will be unable to do that with this. Yeah, I think it should allow me, right? 2.2, right, okay, right here. So I'm follow this line straight across, okay? My hand is not so straight. And it hits the graph right here. So I'm going to come right down, okay? I'm going in the center of my line. My line is kind of bold, but I want to see exactly what I'm showing you right here. If you subdivide the lines based on how I scale them, right? Because moving by 5, so each line is a 0.25 right here, right? So again, if you check the lines, you realize you get right here is 13, okay? So you could just because from 10 to 15 is 5 and it's 10 lines, so divide by 5, so each line is 0.5, right? All right, so now we could just jump on to the answer. So the answer here is 13 based on our graph. So get 13 here. Okay, so the concentration it will be 13. Okay. So on 13%, because the concentration is measured in percentage. So let's put percentage right there, right? So 13% is the answer there. Our last question for this question, or part of this question, is to list one other factor that can alter the rate of an enzyme control reaction. And this one was concentration, right? So beside concentration, what else could cause the reaction rate to change of catalase? So other things that may affect catalase to react is pH. And notice how pH is written lowercase p, uppercase h. And beside pH, we also could have temperature, okay? The temperature could also affect the rate of how enzymes work. Okay, so those are the two other factors that we can have there. All right, so let's move on to our next question. All right, we're moving a little bit fast, which is great, which is absolutely awesome. So this is question number 4 here. And for question number 4, it reads that diagram A shows a cell during a stage of mitosis. All right, and what mitosis is produces the same number of chromosomes, only 2 daughter cells, and so on. All right, so now we said, let's go to the first part of the question. So we have diagram A, diagram B is empty. It said, name the stage of mitosis shown in diagram A. Now, how we know what diagram A is? If you notice the chromosomes, they are aligned in the middle, which is on the metaphase plate. And so since the chromosomes are aligned in the middle, then we're going to give this our metaphase. And so please remember, just to put it, let me put it right here for you real quick, right? The phase that you need to remember is PMAT, okay? PMAT, which means the prophase. So let's put PMAT right here just to kind of remind you. So prophase, metaphase, anaphase, telophase, right? Before the prophase, you have interphase, right? Which is no division at all taking place there. And then after the telophase, you have cytokinesis, which only the cytoplasm will divide. But prophase, metaphase, anaphase, and telophase is the nucleus is divided, so we call it nuclear division. All right. So anyway, let's move on real quick here, and this is called the metaphase. Metaphase because it is aligned in the middle. So you can remember that M for middle, metaphase. Okay. All right. So let's go to the next part of the question right here, and the next part of the question state complete diagram B to illustrate the next stage of mitosis. Make sure to label the spindle fibers, the centrioles, and chromatids. All right, so the next stage after the metaphase will be the anaphase. And you can remember A for anaphase, which means they go to opposite. They're kind of antisocial. Do not use the word antisocial in the exam though, but that's a way to remember it, right? So the prophase is when they press together. Don't use the word press. It actually condenses the correct word. Metaphase, middle. So P for press, they press in the middle. M from middle, which is line up in the middle. A is antisocial, move to opposite sides. We do not use the word antisocial though. Just say that the chromosomes move to opposite sides of the cell or opposite poles of the cell. And then the telophase is when you have the cleavage furrow forming and the nuclear membrane start to reform. All right, let's jump into this and answer this directly, okay? So now we're going to split these up, okay, and let them be on either side. So we're going to have, from the first one, we have that, okay? I'm just going to make them a little scribbly, you know, to make them look really nice. Okay, and then we have that. Okay, so it's 4 of them. Okay, so that one, that, and last one. Okay, great. So again now, let me change this into a different color. So I don't want to mix up everything here. Let's put this in red. And so attached to here, we have what they call the centriole. So I'm going to put the centriole right here. And the centriole is at right angles to each other. The structure is at right angles to each other. So I'll put it like this, okay? And then now, attached from the centriole to the chromatids, we are going to have our, let's see what I could use there. Let me use black, yeah, nice color. So here now coming from, oh no, I don't want that. I want my pencil to be in black. Okay. All right, awesome. So coming from this, we'll have this come here. All right, so those are the spindle fiber and joining. So you draw them like that. And you're going to label them. So they say label them. All right, so that there. One come here. All right, one also here as well. And so good. Those are our spindle fiber. All right, so let's just put the names in real quick. So in terms of labeling, let's label them real fast. All right, so I'm going to put a line so I could label it on one side here. Let me just write the names in first. Makes sense? So I'm going to put chromosome. So I'm going to label our chromosome. And put chromosome here so we could see it eventually. Okay, so chromosome. And this, we should also label our spindle fiber. So I could put spindle fiber right here. Okay. Spindle fiber. Right. And we also have our centriole. All right, so they say label those 3. Centriole. All right, so great. All right, so now let me just quickly attach what is what to what. And so let's use probably red, okay, to line up our things. All right, so yeah, our centriole, which is these red structures that I put here in 90 degrees to each other. So those are the centriole, those are the red structure. All right, so this is centriole. Spindle fiber will be the lines that connect the centriole to the chromatids. Okay, I'm going to change this. I write chromosome, I should put have chromatids. Okay, so I'm going to change that into chromatids. Not chromosome, but chromatids. All right, in the previous diagram, it would have been chromosome, but for this diagram here, you're going to be chromatids. Okay, so let me quickly change that. I want to get into the box. Okay, great. All right, so chromatids. All right, chromatids. All right, TID. Okay, so there's 1. All right, so that would be our labelings, right? So a chromatid, or a spindle fiber, or a centrioles. All right, great. All right, so let's go back to the next part of the question now, and it states that explain how mitosis ensures that a new cell is genetically identical to its parent cell. So in other words, how the cell remains to be the same exact thing. All right, so here now, the first thing to remember is that the bottom line is it produces the same number of chromosomes, okay? So what I want to put here, just kind of guide you a little bit, is that the same number of chromosomes are produced, okay? Let's put it right here. The same number of chromosomes are produced, okay? So that's the bottom line we should include in this. We know that the number of chromosomes will be the same, but how this is achieved? How this is achieved? That's a thing now. How is it achieved? So firstly, let's talk about what happened here. Is that before you have the prophase, metaphase, anaphase, and telophase, what happened before mitosis is what you call the interphase. So what I have now is that they're replicated. I'm going to put there where they're replicated, and this is coming from the interphase. So the replicated chromosomes align in the middle, right? Align in the middle of the cell, and this is actually in metaphase, right? So after the prophase and metaphase align in the middle, I'm just kind of going to, this is only 1 point, so I just want to make it as briefly as possible, right? Align in the middle, of the cell, line in the middle of a cell, in a single file. So they're aligned in a single file, right? In a single file. And because in a single file, right? In a single file, so that each new cell, all right, will receive or have each new cell receive, okay, I make a mistake here. Okay, receive the same number of chromosome. Okay. All right, so that's the bottom line there. That's what happened there. So the replicated chromosome, they are in the middle of a cell in a single file so that each new cell receives the same number of chromosome. Okay. So the same number of chromosome will be in the daughter cell compared to the parent cell. So the parent cell has the same number of chromosome as the daughter cells. Okay. That's the bottom line. So the same number of chromosomes are produced in the daughter cell as to the parent cell. Okay, so parent cell, daughter cell, they have the same number of chromosomes. All right, so for part D for this question, it said, if a cell has 18 chromosomes, how many chromosomes will each of its daughter cells have after mitosis? Now remember, mitosis, as we established just now, that produces the same number of chromosomes. So get the same number of chromosomes, which is 18. If it's meiosis, then it'll be half that number of chromosomes. It'll be 9, okay? Because meiosis produces haploid cells, while mitosis produces diploid cells, okay? Let's jump on to the other part of the question here. This one is to complete the table to show 3 differences other than the effects on chromosome number between mitosis and meiosis. So we can't talk about the number of chromosomes in this one, right? And so for this one here, for mitosis, it is 1 division. So we're going to have 1 division in this one. Okay, so 1 division. And for meiosis, we'll have 2 divisions, okay? And remember, comparing the same thing at every time they ask for differences, please compare the same thing or the same feature. Do not mix them up all over the place, right? And for this one, for mitosis, we'll have 2 daughter cells. Okay, so we have 2 daughter cells. And for meiosis, we'll produce 4 daughter cells. Okay, so we have 4 daughter cells. All right. And for mitosis, as a result of the type of processes taking place, no crossing over, there is no variation in the cells. That means the daughter cells will be exactly as the parent cells. So no variation or no genetic variation, okay? So it results in no variation. But for meiosis, results in variation. So there will be variation within the parent cell and the daughter cells, okay? So you have a variation in this. And the variation is because of crossing over and synapses taking place, okay? And that is happening in prophase 1. All right, so here we finish with this part of the question. Now we're going to go to section B. Again, please read through the entire questions, the 3 questions, and choose the best 2 questions. All right, I have already gone through this and I've typed it up and so on. So I'm going to jump into it. But this first part of question, number 5, is a long question. But the good thing with long questions, they're in small pieces. So even if you lose a mark, it's only a mark rather than losing 5 and 6 marks at once, right? So sometimes it's good to have long questions. Sometimes it's not good because sometimes it really drag your memory and kind of asking a lot of different stuff. But again, you choose what is most appropriate for you, okay? Or what works for you. So let's go to the next part of the question. All right, so now we're in section B. All right, and again, you only answered 2 questions out of the 3, but I'm going through all 3 questions for you right now. All right, so for question number 5 is really a long question, right? Really long. All right, but again, it could be a sweet question based on the breaking down of the points, which sometimes work really well. All right, so question number 5 is to define the following terms. I'm looking for deforestation and conservation. So for deforestation is simply the act of removing trees or clearing forestry or forestry areas on a wide scale generally to a point of having little to no trees or forestry. Now, conservation, it is a set of planned actions used to protect, maintain, and manage resources that will prevent them from being or from becoming extinct. So you want to make sure that you preserve whatever you're trying to conserve. All right, so it could be natural resources, it could be animals, it could be plants, it could be an area. All right, so you conserve these things to make them be lasting for generation and generation to come. Now for part B, it's a list 3 reasons for deforestation. And so deforestation could be done for a number of reasons. I have a number of lists right here. So one, it could be for agricultural purposes, for planting different things. So you take down all the forests, you take down all the trees, and you can clear a whole land or entire area just to rear livestock. So you could have livestock, which is animals, or you could simply plant vegetables. So you take down a whole forest to plant vegetables and so on. Again, you can clear a whole land or an entire area for lumber, to make paper, for cosmetic purposes, to make furniture, and all these different things, right? And for charcoal, again, charcoal production, construction, to make roads, to make factories, or to even build cities. So there are a number of reasons that we may do deforestation. So next part of the question here, right here, is to explain how deforestation can increase the amount of carbon dioxide in the atmosphere. And so the first thing you need to remember is that photosynthesis, right, is a main, okay? I've maintained right here. It is a main process, okay? So it is a main process. Not maintain, is a main process. Let me get this off. Okay, so it's a main process. Not maintain, is a main process. All right, that removes carbon dioxide from the atmosphere. Okay, so bottom line is the main process that takes away carbon dioxide from the atmosphere. And so removing the plants, so when plants are removed by deforestation, then the rate of reducing carbon dioxide is lowered. As a result, more carbon dioxide will be in the atmosphere. Another way to say, this is in red, is that the rate of releasing carbon dioxide in the atmosphere by respiration is greater than the rate of removing carbon dioxide by photosynthesis. I kind of love this last part right here. I really love this part, right? Because remember, organisms respire, release carbon dioxide. So putting carbon dioxide in the atmosphere and taking them back out by plants, which are not there anymore, of course, increase the amount of carbon dioxide in the atmosphere, right? All right, so part 2 of this. Again, I'm trying to move real fast because I know time is up on me. Now, this part is said, explain how an increase in the amount of carbon dioxide in the atmosphere can harm the environment, and a number of reasons can be given here as well. And so 1, carbon dioxide in the atmosphere could cause climate change or global warming. And simply put, because carbon dioxide will create what they call a greenhouse effect. And so it results in warmer temperatures, and rapid change in climatic condition can lead to migration of species. So it can harm the environment. For 1, it could create or cause higher temperatures and rapid change in weather or climate. And so because of this reason now, you have a migration of species. Species cannot live in these areas anymore. They cannot tolerate these changes. Again, because of global warming or the greenhouse effect or warmer temperatures, you may have a rise in sea levels. And of course, rising sea levels could be simply because the ice cap is melting. So even though the ice cap is melting miles away from us, eventually water may go into the ocean, and the ocean level rise and eventually affect small islands. And sometimes, and if it rises so high, some islands may disappear eventually. All right, so that could be really very, very detrimental. Other reasons may include acid rain and ocean acidity. All right, so because the carbon dioxide could mix with the rainwater or the precipitate, and then deposit hydrogen ions, and then the water become acidic. And because of this acidic nature of the rain or precipitation, then it may damage plants, may damage building, may deplete nutrients in the soil, all right, and also make the ocean becoming acidic. And they know the bad nature of that. If the ocean becomes acidic, then yes, coral reef can be damaged. Okay, ecosystem could be destroyed. Food sources could be lost as well, and so that could cause a great chain reaction and so on. All right. So now part 3 of this is state, describe 2 social impacts of deforestation. So 2 social impacts, and so it could be 4 marks to state the social impact and describe them. That's 2 marks for each. Okay, so I think I have more than 2 here anyhow. So one of them could be loss of culture. And how deforestation could be causing loss of culture or indigenous people is simply because some people or cultures depend heavily on the forest for food, medicine, shelter, and other practices. Because, of course, some culture even worship in the forest as well, right? And they have specific sites that they go to worship and all those things. And certain traditional food that they cook or come from the forest, like certain plant materials that they use. So definitely could affect culture and even affect the indigenous people who live there as well. So loss of farmland, okay? And if you lose your farmland, if farming is a main source of income, and imagine you lose those land, the land is no longer fertile, or they build cities there and you have no work, for example, or no means to get your food, then this could lead to starvation or social unrest, right? People could get real upset and protest, and all those type of things could be going on. All right. And next could be an increase in crime or corruption. And this is simply it, because deforestation is a business, right? And of course, once money is involved, then person can become greedy and engage in illegal mining and also in other illegal activities. As a matter of fact, they can even start to kill each other over the money made, because people may think that they should get more, or they're not earning enough, or they want to make millions out of it, and so they could cause a lot of problems, right? And next one could be exploitation or cheap labor or child labor. And of course, because sometimes around these forestry areas, you have poor people, or you may have a higher rate of poverty being experienced or seen in these areas because of lack of amenities and so on. And so the operators of these operations or deforestation processes, they can exploit persons living in and around the forest areas, right? So that, again, could cause another social problem, all right? Now, part D is that deforestation causes loss of biodiversity. What is biodiversity? And for one mark question, it said biodiversity refers to the variation of species or organisms living and interacting in a given ecosystem. Okay, so it's a great number of differences between species, different type of species, different types of organism, just living in one area. All right. Now, part 2 now asks us to explain how deforestation causes loss of biodiversity, and some of the reasons we mentioned up top will come back here to help us again. And this one is saying that due to loss of resources and food, organisms may migrate or become extinct due to destruction of habitat. All right, so organisms become vulnerable to predators. So destruction of habitat, organisms become more vulnerable because they're more exposed, and so on. And because also of destruction of habitat, organism may also migrate. Okay, because again, nowhere to hide, they're eating easy and so on. Okay, all right. So those are some of the things that definitely that you might see. All right, so let's jump on to the other part of the question right here. Now, this part is a loss of nesting areas as well, okay? Loss of nesting areas. So deforestation could because they take away the trees, birds may have nowhere to make their nest, okay? And even some other organisms may even make nests in the roots of trees as well, and even in dry barks and so on. And so loss of nesting areas for birds can reduce the variety of birds in that ecosystem, okay? Because if they have no way to make nests, then they're going to go somewhere else, right? Climate change as well, all right? And the inability for organisms to adapt may also lead to extinction. So when climate is changing because of deforestation, due to global warming, because of more carbon dioxide in the atmosphere, then again, if organisms can't adapt to these things, then they can become extinct, especially if they can't migrate, okay? So those are some of the reasons there. And we're still at question 5. You know, question 5 is really a long question, isn't it? All right, so part E of this, now we said, suggest 2 actions that we can take to try to reduce the loss of pine forest in the Bahamas. And this is for 2 marks. And so one of the answer here could be replanting trees or reforestation, which is reforestation, the planting trees or replant the trees. All right. You can use less products that come from pine trees. Okay. So in other words, now if the products are not selling, no one will go to take out these trees out of their areas, right? Okay. So that's another one there. All right, so remove invasive species such as the casarina. invasive species could really cause a damper on the natural organisms or natural plants that grow there. All right, and so removing the invasive species will make the ecosystem flourish. We can also establish laws to protect the pine forest. We also could engage in educational programs that will assist persons to understand the benefit of the pine forest, and then in return, they will want to help to protect it as well. Because sometimes if you don't know, lack of knowledge could be a really serious thing. But once you have the knowledge, then you're able to have that power to do the right thing. All right, so number 6. There's a list of the elements normally present in proteins. And so the elements that are present in proteins will be carbon, hydrogen, oxygen, and nitrogen. Again, sometimes we may have phosphorus and sulfur, right? But these are the elements present. Again, I give you the name and the symbols, okay? All right, so let's jump on to the next part of the question right here. For this part, it said describe how you would show that egg white contains protein. So you're doing a protein test. First, you have to remember what do you use to test for protein, all right? So first, get the egg white, all right? So you add the egg white to a test tube. Then you pour some birth solution into that test tube. And again, you want to do it sparingly to make sure that at least you cover the egg white itself. Point to note is that the egg white, the birth solution is blue in color, so you need to know that, okay? And again, what you'll do is give a shake, and you put the test tube with the solution containing egg white to sit for a while. All right, you'll make that sit for a few minutes. So you're going to observe a test tube after a few minutes, and then if the solution turns purple or violet, then you know that protein is present. Again, remember, it is the birth solution changing color, not the egg white, okay? So please note those small things. They can mean well in exams, all right? Now, part C of this question, it said, name 3 substances found in living organisms which are made of proteins. So there are a number of things present in organisms that contain proteins, such as hormones, our gametes, which are sex cells, eggs and sperms, blood cells, enzymes, antibodies, fibrinogen, and not just only fibrinogen, because fibrin as well, right? Thrombin, prothrombin, they are all proteins that are found in the blood, in the blood that will assist in blood clotting, okay? Our muscles are made up of protein, all right? All right, and not just only that, we have our fingernails also made up of proteins, our hair made of proteins, right? So we have proteins in many different structures inside of our bodies. All right, part D. It said discuss the role that 2 named type of microorganisms play in food production, and this is for 6 mark. And so here I kind of break it down different ways. So I'm going to show you something real quick. And so one of those organisms is a fungi called yeast, okay? And yeast can be used in more than one ways. And so we can have yeast in the baking industry or the brewing industry. All right, so the brewery industry or the baking industry, yes. And so in the baking industry, yeast is used by allowing yeast to respire anaerobically. And when they respire anaerobically, they produce carbon dioxide that cause bread or dough to rise, okay? And when the carbon dioxide rises through the bread or through the dough, it leaves those pockets or whole pockets or air pockets. And because of that, the bread itself or any dough or products that make from dough then become soft to give that soft characteristic of bread, okay? So it's very useful in making bread really soft and nice, all right? In the brewing industry or alcohol production, then yeast is also respire anaerobically. And by using the glucose in selected fruits or products that make specific types of alcohol beverages. So if you want to make wine, for example, you may use some grapes or you may use some strawberries. So different fruits may give a different flavor. But the bottom line is that the yeast will respire using the sugar in those fruits, all right, and to produce alcohol as a result. And of course, the benefit of that is that these distilleries will distill the alcohol from what is being fermented. All right, so it's a fermentation process. You can make a note of that. And so they distill the alcohol from it to make it safer because the residue from the process may not be so good for consumption, right? All right, so great. All right, again, the next organism here or microorganism here is a bacteria. So bacteria used in the production of many food products such as cheese, yogurt, vinegar, and soy sauce. All right, and so bacteria They are generally responsible for the fermentation process in the above productions, which is cheese, yogurt, and all these things, right? And so for cheese to be specific, in terms of cheese, the bacteria itself, right, will cause spoilage in the milk, resulting in the separation of what they call whey and curds. Now, the whey is the liquid part of the milk, and the curds will be the solid parts of the milk. And just to give a little summary, what happened in the process of cheese making, the milk is being used, the bacteria is involved, and then it separates into whey and curds. The curd is what is needed to make cheese. Then after the curd is formed, the next set of bacteria is used in the formation of the cheese. So there's a forming process called make forms. And then another salt-tolerant bacteria again is used in the salting process. And then the next bacteria is used again in the aging process. So bacteria is used right out through the cheese making process, all right? So these are just the stages or the steps in cheese making process. That's just additional information for you anyhow, right? All right, so part E. It said, describe how pregnant woman supplies her developing baby with protein. So I want to look at how this pregnant woman can supply the babies with protein. Again, baby do not digest. The fetus do not digest. Okay, they do not release waste in like feces. Okay, they do not do those things. All right, so just to make a point of that. So how they get their food and how they get rid of the waste is through the mothers by the umbilical cord. And in the umbilical cord, you have the umbilical artery and the umbilical vein, and they are attached to the placenta, not directly, but because of the membrane of the umbilical cord. And so things only move across the mother's blood to the fetus blood by diffusion. So let's jump into the first part right here, right? So how to outline this to say that the mother now eats food containing protein, such as meat, nuts, grains, and all those things, right? So any protein containing food, she will eat those. And then now she will digest the protein using enzymes to get polypeptides and peptides. And then eventually the polypeptides and peptides break down into amino acids, which are the end products of digestion within the mother. And then now the amino acids are absorbed into the mother's bloodstream and then sent to the placenta, where now in the placenta now the amino acids will diffuse across the placenta into the fetus blood, right? And the fetus blood, they are traveling again, as I mentioned, in what they call the umbilical artery and the umbilical vein within the umbilical cord, okay? And then now the amino acids eventually travels through the fetus and into the tissues and into the cells where they are assimilated. So they become assimilated in the baby to make new blood, to make new cells for growth and development, right? And that's how the mother supply her fetus or unborn baby with proteins, okay, in the form of amino acids. And then they convert into protein within the fetus itself. All right, last question here, and we're on our roll. All right, so number 7. It said, name the 3 main parts of the human brain and describe one general function of each part. This is a 6-mark question. Relatively easy question, isn't it? Because you get this mark so easy. Now, cerebrum controls memory, control voluntary actions such as walking, talking, and so on. But how would I write this though? If I would change this a little bit and put it controls voluntary actions. That's it. So cerebrum controls voluntary actions, all right, such as walking, talking, intelligence, and so on. But they're also responsible for our emotions, also responsible for our memory, all right, and intelligence. Like what you're doing now, you're taking time out to consciously study. So yeah, you are using your cerebrum to be more intelligent. Absolutely. All right. Anyhow, cerebellum. Cerebellum is used to control balance and coordination, right? And so if you're in an accident, for example, God forbid, and you damage your cerebellum, then you lose balance and coordination, right? Or if you drink alcohol beverages, which you should not be drinking anyways, and if you do that and you become a little bit unstable, you lose coordination, you lose your balance. And now your medulla oblongata controls the involuntary actions such as your heartbeat, digestions, and so on. Those things that you cannot control is controlled by your medulla oblongata. All right, so let's jump to our next part of the question right here. Part B, it said list the structures of the eye in the correct order through which light ray passes on its way to the retina. 4 marks. And just bear with me right here. The parts in blue, which will be definitely the things that we want, definitely, right? And so part in red, I just put that there as extra because the aqueous humor and the vitreous humor are not structured. They are substances. It's a liquid portion, right? It's a liquid fluid, all right, or watery fluid. And so cornea will be the first part, and then it travels through the aqueous humor, then it goes to the pupil, then it goes to the lens, then through the vitreous humor onto the retina. Okay, so that's the pathway that light will take into the eye. Notice I didn't list iris, right? Because the iris is not transparent, okay? So you go between the iris, which is the pupil, okay? So just to make a note of that, all right? Because light must travel through transparent objects. All right, so let's look at part C. Part C now, it asks us to describe with the aid of diagrams how ciliary muscles and the suspensory ligaments work to change the shape of lens to focus on near and also distant objects. This is a whopping 10-mark question. Woo! All right. Now, how do we approach this? Quite easily. I like this question. It's short and you can get the mark so easily. All right. But anyhow, let's go into that. All right. Don't want to waste too much time. All right. So first and foremost, what I want to do is compare them with you in terms of the diagrams. 10 marks. So I would suspect that at least the diagrams earn about 2 marks each, or at least 3 marks each. They're about, right? So let's say 2 marks each. So what I wanted to do is always to label diagrams. Please do not draw a diagram without labeling or annotate a diagram. Say something about the diagram. Do not leave diagrams, especially see these letter marks. Do not leave them unlabeled. Please do not do that. So for near objects, just let's point out the labelings before even go into what is happening here. So we have the ciliary muscle, all right? We have the suspensory ligament, and we have a lens, all right? Ignore my bad drawings, but, you know, just to depict the information here. All right, so let's compare the two lens within far and near objects. So for far object, notice the lens itself. Notice the ciliary muscle and the suspensory ligament, right? I should have them side by side, make you see, but let me go into it a little bit, and you could see them, and then I go into the answer, right? So notice the big difference, right, between the two. For near object, the lens is shorter and thicker. In far object, the lens is longer and narrow, okay? So let's go to what is happening that causing those changes. So for near objects, right, for near objects, this is what is happening here for near objects, is that the ciliary muscles, which is this part, they will contract, okay? And then now the suspensory ligaments, they will relax. So notice they are longer compared to the next diagram I'm going to go to. The next diagram, they will be shorter, which means they are contracting, right? And so because of this, the lens thickens and become rounder. And this now will cause the light ray to refract onto the retina properly, okay? And so for far objects, notice there's the opposite for what we did above. So for the far object, again, there's no need to label it twice. All right, if you have time, you want to go ahead and do it, that's also fine. But there's no need. Once I label one diagram with the same structures, there's no need. All right, so for far objects, what is happening here? The ciliary muscles, they relax, while the suspensory ligaments contract, which means they get a little bit shorter, and these get a little bit longer, okay? And so as a result of that, the lens become longer and also flatter, okay? And so that will be the exact thing that is happening there. So again, the notes, 6 marks, test is 3 points each. And so we get 4 marks from both diagrams, 2 marks each, include the labeling. All right, so this is where we actually end this review. And so again, I'll see you in the next review. I'm trying to get a next review in today. Hopefully I can. And then, you know, we do some paper 5 possibly. I don't know, but we'll see how the time goes. So see you soon.